In this video, I'll talk through the 2025 Higher Biology paper and explain how I would approach each question. Starting off with the multiple choice in question number 1, we're asked about the DNA in various parts of cells. In the nucleus, that's where we have the linear chromosomes. That's going to take us to be in either A or D. Chloroplasts have circular chromosomes, and then mitochondria also have circular chromosomes. That makes our correct answer for number 1, answer D. If we're asked about a prokaryote, like a bacteria, that would be circular and plasmids. And yeasts have linear in the nucleus, circular in the mitochondria, and they also have plasmids, depending on the wording of the question. In this next question, we're asked about the tRNA involved in translation of the mRNA transcribed from this sequence. A lot of bits going on there. Right, this is your DNA. We could then take our time going through. Well, mRNA, we're going to take any C's, I'm going to turn into G's. A is going to bind with U when it's an RNA. G is C. I'll go through the rest of them. And then when it then comes to the tRNA, then we're going to flip it back. And we have got our 4 anticodons. Only 2 of them have got a single uracil in them. Correct answer there would be B. When we're jumping from DNA to tRNA, what we can do is just, it's going to be the same code, except from any T's are replaced by U's. So you see that first one is exactly the same, but anything with a T then becomes a U. It can be a kind of shortcut. But as we did the first way around, that would flip it into mRNA and then into tRNA, depending on the question. Next one, we're talking about validity of an experiment. This is where we're looking to control all controllable variables. We're trying to make it as fair a test as we can. Well, different fruits isn't going to be making it more valid. Using a balance to measure the mass of the DNA is to do with the outcome at the end. That's what they're measuring. That's the dependent variable. We're not going to be doing that as a fair test. This is about, for validity, we're talking about setting up the experiment and making sure it's running in the same conditions. Which the water bath keeps the temperature fairly constant, depending on how good your water bath is, but it will keep it more equal throughout. Repeating is to do with reliability. If you've had any rogue or anomalous results from that, that's looking like C is the one I would pick from there. Next one, we've got mutations making different proteins. Comparing, right, smaller proteins travel further in the gel. Okay, which is one diagram shows of these proteins. Protein X is your normal protein, which means it would be the expected size that it's supposed to be. In a miss sense, we're swapping one amino acid for a different amino acid. That means that from a size perspective, they'll be very similar, if not the same size. And then in a nonsense, it's a premature stop codon, meaning you get a shorter non-functional protein. That will mean that one will be smaller, and smaller proteins travel further in the gel. So I'm looking for one where X and Y would be the same distance because they're same or very similar sizes. That knocks it down for me to one of the top 2 answers, A or B. Z will go further. The proteins are put at the top part here and are travelling down the way. In A, protein Z has travelled further, showing that it's the smaller protein, making A my chosen answer here. Next one, we're talking about gene transfer. Okay, so we have different ways of passing on genes. Horizontal is within the same generation. And the first one, it's not. It's going from the mummy and daddy hamsters to the children. Asexual. In number 2, the difference is here where it's a clone of the original yeast, but it's still going from the parents onto the offspring, which would make that vertical, not horizontal. Here they're saying the words that I was looking for. Same generation. Number 3 is our correct answer, and B is the answer we would choose. On to C, pharmacogenetics is to do with medicines. The pharmacy is where you can go to pick up medicines. If you call it the chemist, then that makes it a bit more difficult to make that connection. Is there anything that says anything about medicines? Drugs, medicines, using an individual's genome sequence to select the most effective drugs. That looks good. Which looks good to me. What would the other ones be? This would be bioinformatics. I remember that as in school, you might have or had an IT class looking at using computers and software and things like that as well. So computers, IT makes sense to me. Evolutionary, we're into phylogenetics, tracking the development of different species over time. Descendant, so if we're talking about the genomic sequencing, just looking to derive some information about rates of diseases and things like that as they go through. Next one, we're talking about types of proteins embedded into membranes. Are there ones that are not? Okay, let's try and work our way through these. So pores are openings that things can make their way through. That is, histone is attached. Histones are related to DNA in the chromosomes, so it's not going to be that one. There are enzymes, but it's not going to be that. I can again get rid of that one. I can again get rid of that one. So actually, the only one that works is going to be C. Now, the list that you might have memorized, there are pores, enzymes, and pumps. Channels, this is sometimes referred to as a channel. It's an opening that things can pass through. What they've tried to do to trick us here is they have given us a specific enzyme in ATP synthase, which is responsible for making ATP. It is a type of enzyme, and they've just tried to hide it a little bit, because I remember that histones are a protein that are associated with DNA. I can rule out those ones, and that has flagged up the correct answer. Next up, we're talking about glycolysis. Glycolysis is the section that does not need oxygen. It can happen even in fermentation conditions. How does that help me? Well, I can get rid of B as an answer. Okay, so if I look at C and D, well, glucose is broken down into pyruvate. So you don't need pyruvate, you make pyruvate. I'll chop out that one. Do we also need NADH? Well, NADH is made. The hydrogens are removed by the dehydrogenase enzymes, and then the hydrogens are passed to NAD to make NADH, which moves on to the next section. So although the glucose is correct, we don't need NADH, we make it. That means the answer is going to be A. NAD is needed to grab onto the hydrogens and the high energy electrons to take them forward. ATP is broken down in the energy investment stage, and then you get more ATP out in the energy payoff stage. A is what I'm going for there. Next one, we're talking about reliability. Mentioned this earlier. We are talking about repeats. Doing the experiment more times, and then taking averages and looking at those things as well. Okay, so using a control, this will allow a valid comparison to be made. Gives us a baseline to compare our results to. Carry out experiment at each inhibitor concentration. That is textbook what I was saying. Repeating it. Wider range lets you see more of what the actual trend is. So it gives you more information about the trend. And then this is going to increase the validity if you keep everything else the same. In this one, we're asked about the ability of an organism to maintain its metabolic rate affected by. So what things can affect the metabolic rates? Well, temperature is going to be one of them. Abiotic is to do with non-living factors. That will maybe help. Well, disease and predation are to do with living factors. Not that, not that. We are down to 2. What's the difference? So we have temperature. This is to do with the salt content of it. And this is to do with other microorganisms. So now I'm saying there, well, other microorganisms would be biotic. They're living. So it won't be that one. So A is the one that's got 3 non-living factors. That should be it. Next one, we've got an increase in body temperature. So if someone gets really warm, their blood will rush to the skin to help heat to be lost as radiation. So radiate off and cool down. Right, so how is information sent to the effectors? So the effectors are part of the nervous system. So I'm going down into these ones. So it'll pass through your sensory intermotor neurons. We want more blood to rush to the skin to cool off. That means we want to be opening up the blood vessels. We want to dilate them. And then constriction is when they tighten up and it reduces the blood flow. That's leading me towards D as our correct answer. If you're struggling with those two, I think of the snake, the boa constrictor that squeezes on their prey to squish them. Moving on. Right, next. Compare these data sets. What is supported by the data? Okay, so lactose used to break it down, timed over time. Okay. Glucose production is faster in human milk. Well, if we take at every point going down it, we have started at the same level and we're getting more of the glucose comparatively in the human versus the cow milk. That sounds good to me. Glucose reaches its maximum concentration sooner in human milk. Well, it got to the maximum somewhere between 2 and 2.5 minutes, whereas for the cow milk, they reached a peak earlier, so that's not going to be that one. No lactose remained in cow milk in 1.5 minutes. Well, if that was true, that would be your final result, but we can see here that it's still going up, so that's not going to be that one. Maximum at 2 minutes. Well, it didn't reach a maximum in the human milk because the human milk was still going up past 2 minutes. So yeah, I'm comfortable with that one. Next one, regulators and conformers. A conformer will change its internal conditions with the external conditions. A regulator will try and keep their levels at a set point or within a small band, a range. Right, let's see then. So I am looking for regulators to be flat. That's B or D. Have the dashed line going flat. If the temperature was increasing, the body temperature of the organism would increase as well. So it would slope up the way. Here, what's happening as it gets warmer outside, they're getting colder. That's not what we are looking for. D is going to be correct for number 13. Moving on to this, we're talking about migration. That's when they're running away, flying, walking, running away to a different area to avoid the adverse conditions. And then survival ones would be things like hibernation, estivation, daily torpor. All let them survive in that environment. They can survive those conditions. Next one, we have got our graph of time and number of viable bacteria. Okay, percentage of bacteria killed by 5% after 50 minutes. So 5% is the full line. So from 50, we'll go up. And we'll run that along to there. Okay, right. What does that tell us? That tells us that 10 times 10 to the power of 9 cells per centimeter cubed were alive. And we started with 40 times 10 to the power of 9. Importantly, we have to work out how many died. So if you started with 40 times 10 to the power of 9 and you have got 10 times 10 to the power of 9 left, that means that 30 times 10 to the power of 9 were killed. We have to turn this into a percentage. That's like when you get a test score, where you do how many you got correct divided by how many in total times 100. So in this one, it's how many fall into our category. That's 30 times 10 to the power of 9 over, well, there were 40 times 10 to the power of 9, and then times that by 100. 3 over 4 will be 75%. You can just use your calculator and that will do that for you. This answer is what you'd get if you used just the number from the graph. Checking the labels is very helpful because you can get that that is not what we're to do. Next up, recombinant DNA, which is not correct. Well, plasmids transfer the information. So plasmids do move genetic information about. Ligase is to do with sticking things together, not opening them. That is incorrect. So the cutting is done by endonuclease, which isn't what we've got here. Using the bacteria may result in proteins that are not going to work. They have been folded incorrectly. That is a true statement. Chromosomes, artificial chromosomes, are larger than the plasmid, so you can put in more DNA into it. Ligase job is to stick the desired gene into the plasmid to make the modified plasmid to then go on and be used and be grown and be made copies of B. Wrong enzyme. Right, next up, we have now got these ones. So they have marked out the restriction sites, which is where the endonuclease enzyme will chop out the genes. Transferred bacteria were resistant to ampicillin. Okay. Which feature of the modified plasmid is responsible for this resistance? Okay, so what are the jobs of the different parts? Regulatory sequences, that's to do with controlling gene expression. That makes sure that the gene can start being expressed into proteins. The gene is the desired gene, is the human growth hormone. That's what's getting moved into it as well. The original replication allows the plasmid to be replicated. Then the marker gene gives an advantage to the host cell. Well, the advantage here is that if there's an antibiotic present, it's resistant to it. It will be able to survive and grow the modified bacteria, not the other ones. Next one, they're looking at details of the carbon fixation stage in photosynthesis. Job 1 is find something catalyzed by rubisco. Well, that's involving RUBP. The naming here is quite helpful because that's the ones that go together. That's going to make it number 1 is catalyzed by that enzyme. So not either of those ones. Requires hydrogen ions. In this, for this first statement, the rubisco helps catalyze the RUBP join on to carbon dioxide. The copattern there. There's not hydrogen getting added at that point. After the 3PG has been made, it then requires hydrogen added to it, which then converts it into G3P, making the correct answer here B for this one. I was having a little internal debate thinking about, well, would they really give us one that has the same correct answer, both in one? Now, it could have been if they'd worded it in a certain way. So if it required carbon dioxide, then it would have been for there. But the first one, particularly when it's rubisco, that's to do with RUBP and the carbon dioxide coming together. The name of the enzyme is kind of telling us that what it does for that one. And then the other one is the second part of it. That then turns into the G3P. And then those combine to make the glucose at the end of the process. That one right here, we're talking about pigments. Which pigment is xanthophyll? Right, we're dealing with RF values. So that's the distance by the pigment over the distance by the solvent. Now, we can use a ruler. I'm going to use this one here. So, based on this, my measuring tool, I'm getting approximately 60 millimeters for that one. And then we are looking for something that has a value coming out of 0.55. So we could just go through them all and see which one comes out. One of the things that can help narrow it down, I would say, is, well, if it was 0.5, that's halfway, which in this diagram would be at 30. It's 0.55, which means it's past beyond the halfway line. So that one is looking pretty decent to me. And then when you take your calculator and type in, with this on 34 over 60, that comes out as 0.57 is what I've got in there. Now, I've just clocked there that my line isn't quite in the right place. That was 33, which it should be. And that'll give me the exact value we're looking for. There's not other ones that were within a close value of that, so I still would have went for, which one was that? C. Nope, B. Pigment B. Another thing that we could do is we could work backwards. So if RF is the distance of, so pigment over solvent, well then pigment, doing some rearranging, is going to be RF times the solvent, and then that's going to be 0.55 times 60, which gives 33 millimeters. We can then go and look at the one that's at 33 millimeters. Next one, we're talking about a field trial. All right, what's the purpose of each of these things? Randomly is going to prevent or reduce bias. If you had a stake in the game and you were trying to show that your maze was better than other people's maze, and you also know that in your fields there's a certain part where everything grows really well, you might put your one in there deliberately. Or if you know somewhere that's pretty bad growing, you'd put the competitor's one in there perhaps. And the next one, you're looking at comparisons between the different mazes. This lets you see which ones perform better under different conditions. And in this one, we're dealing with repeats. And that's going to increase your reliability. Well, the variability means that there are going to be differences just in general between how much of the crop is made. You're able to get a more representative value for your results rather than just relying on one number. You've got a few numbers you can take an average of and look at. So that's looking like B. Yeah, I'm going to go for B. How many times larger the average area per pack is in region X compared to region Z? Okay, we have total areas, we have number of packs, so we are looking for area per pack. So we have to do the area divided by the number of packs. So 240 divided by 2 is 120. 351 divided by 14 equals... Now I've just clocked, I don't actually need to do this one because it's not asking us about it, but I have committed, so I will finish. 25.1, and then 840 divided by 28 gives us 30. How many times larger? Okay, so X is 120 compared to Y, which is 30. Well, that is a times 4. If you can't see what the connection is, you could look and do 120 over 30 gives you that it's 4 times bigger. C is the correct answer there. That. So a couple of things to watch out for. The units here are not either of the columns. It's a combination. It's using both of them. So we have to do the area per pack and then do the comparison of it to be a bit sneaky. Here, kin selection is between family members closely related. In these relationships, we have altruism is when one benefits and one's harmed. Mutualism, there's a benefit for both of them. In kin selection, they are helping their family members without expecting anything in return. That is going to be altruism. It's all about ensuring that the shared genes are passed on. Okay, putting that together, benefits. So the donor, one that's given away is harmed, and the other one benefits, and it's done without expectation in return. That's going to be C for that one. If it was mutualism, it would need to be benefit, which actually isn't one of the options. The fact that you've got a harm in there would rule out mutualism right from the beginning, regardless of the question. And then, yeah, we'd get down to C for number 22. Moving on, we're talking about a large genetic diverse population. This is feeling awful bottlenecky. We're just a very low number, loss of alleles, and then it recovers, but it's lost a bunch of the genes that were there originally. That's very much feeling like this. Natural selection, survival of the fittest. In B, you're talking about isolation. Starting point for heading towards different species. For C, this would be there's starting to be an increase in the variety of alleles after some sort of catastrophic event. So they're recovering. Here, though, they've recovered without the original alleles, many of the original alleles being present for that. But bottleneck, large starting point, narrows in, you're losing diversity from that. Here, we're talking about species diversity. So how many species? That's the richness. And how many of each species? We're talking about the abundance. So number of different species, that's what I said. Number of frequency of alleles, that sounds like genetic variation, diversity, genetic. We need to at least, so she's got the right, yeah, proportion, how many of each species there are in that environment. That is 1 and 3. That is B as my answer. Moving on to an invasive species. So they have to come from the outside. They've rapidly spread. And they have to harm the other native species. So harm native species. Introduced from outside the UK would make them an introduced species. And they can just peacefully coexist with the natives. Become established, that would then be naturalized, where they're able to then start reproducing in that area. Eliminated native things. That's what I was talking about. And then occupy the same habitat. Well, your naturalized species and your introduced, like your naturalized species also occupy the same, but they don't have the negative effect on the numbers of the native ones. For that, so that should be C for that one. Over to the next one, we're on to DNA into PCR, polymerase chain reaction, where we're making lots of copies of DNA very quickly. We're amplifying the DNA. So at the 3 end of the DNA strand, here's my wonderful diagram. We have the phosphate at the 5 end. We then have our sugar. Not gonna lie, never to put it in, but it doesn't really matter too much. This is the 3 part. Is your sugar, and then you have your base on the end. You get asked about the 3 and the 5 prime ends because those are the parts that join together to make your sugar phosphate backbone. 3 is in the middle from 1 to 5. Sugar's in the middle. 3, phosphate's on the end. That's going to be the phosphate is number 5. So 3 end is going to be your sugar. I would be specific, deoxyribose sugar, because we're talking about DNA. When it comes to the temperatures, I go for all the 5s. So stage 1 can happen between 92 and 98, so I go 95. Stage 2, we're dropping down to between 50 and 65, I go 55. And then up the way, and then the third stage, we're somewhere in between. So between 70 and 80, so I go 75 to give a specific number. It's stating a temperature, it's not asking you for a range. So a single value between those points would pick up the mark for that one. DNA polymerase's job is to replicate the DNA. So it's responsible for the replication. It adds nucleotides to the new strands. Next one, why is ligase not required? Ligase is to join fragments in the lagging strand. And the reason there's no fragments is DNA is replicated continuously. So like the leading strand type thing, it's continuous. It's replicated. Work for that. The specific type of DNA, does it tell us here? Not quite, but it is heat tolerant DNA because normal DNA polymerase would be denatured by the high temperatures. It is not heat tolerant. Next up, percentage of contents that is DNA polymerase. So this is again like your test results. It's the content that is DNA polymerase divided by. So how much is DNA polymerase all over the total volume times 100, which is going to be polymerase is 0.4 all over. Let's get them added together. Which I make to be 25. Remember times by 100. And I get 1.6% as our answer there. And this one, they are asking us how many copies there would be after 8 cycles. 1 cycle doubles number. So we have 70. I mean, to do 8 cycles. So first, second, third, fourth, 5th, 6th, 7th, 8th. Careful here that the cycles are the arrows, not the numbers that we want to put in. So we can just take our calculator and we can just double it every time. 140, that's going to be 280, 560, 1120, 2240, 4480, 889,860, and then 17920, 17,920. There are some questions where they'll give you time, so they might say a cycle takes 5 minutes, you have this many minutes, and you need to do a bit of working out the number of cycles. This one's straightforward. Here are the number of cycles, double the original value that many times. Moving on, we have got things about mutations. Name the type of gene mutation that has occurred in breed B. Compare breed B to breed A and look at where it goes differently. So from here, this is the point where it stopped being the same. The types of mutations that could happen, we could have substitution, where we're swapping one letter for another. But that doesn't explain why the next one doesn't match and the next one. That what it does tell me is one of the frame shift ones. We've either inserted or deleted. Well, you can see that the strand has got smaller. So that feels like we have just got rid of this one. And then they will now match up again. So we have deleted a base and nucleotide. That is the correct answer there. Deletion. Why does that have a massive effect? Well, it's frame shift, which means all codons or all amino acids after the mutation are changed, are affected. And that's why that would work. Why is cattle B going to have an increase in muscle mass? At the top, it tells you it inhibits the growth. So if you don't have that protein, your muscle tissue growth will not be inhibited. It can keep going and keep going. So the inhibition has not happened because you don't have that. The cow doesn't have that protein to stop it. So that is what I would do there. Compare the lengths of pregnancies in beef and cattle. Watch out here that we have got a couple of them kind of separated for that. Okay, so the beef ones are both of the highest values. So beef, cattle have longer length of pregnancy. And then the second part they're asking is about the birth weights. Let's go over birth masses. And this time the two beef ones are the lowest. So dairy have higher, and that would be your 2 points there. Next one, we're asked about isolation barriers. Isolation barriers can be geographical. So this is your physical separations. It could be behavioral, which would be things like their mating rituals. Biological is what they're going to be eating. Things like that about their environment. So there's nothing physically stopping them getting close to each other. It's not with that. It's not to do with what they're eating in the same kind of area. Sounds like behavioral. Probably a nicer, neater way of doing that. Why are so isolation barriers prevent gene flow between the subpopulations, the groups. In this one, we're asked to do a prediction for the next depth. So 60 to 70. I care about the red one, so over on the right hand side. Okay, let's look at how much has been added on each time. So I'll just measure, I would calculate all the differences. So we've had 27 added on and 35. Well, that's gone quite dramatically up. Okay, then we've got, okay, so there has been a dramatic jump in the middle section. As we get down towards the bottom, this seems to be getting what's 200 smaller than the one before it for that. Now, following the pattern, it's looking to me like it will be something in that ballpark figure. So I would probably say it's getting smaller. It got smaller by 200. So if I take another 200 off, I would be wanting to be in that ballpark region, which would mean I would add on 2160 to that last value they give us. And if I do that, that gives 13730. 13730. I've just went and checked the marking scheme to see how big a range they've given us. So we've got 2,000 range to deal with. So anywhere between 12,000 and 14,000. So what we wouldn't want to use is use this one because that would be too small. Nope, that would be far too big and take you outside the range that's possible. We're just looking for whereabouts does that fit in. if all of the jumps had been the same value, you would be expected to give one specific answer. Here, there's a bit of a range because the changes are changing over the course of the table. And that's what we get for that. Suggest a reason why the method used to count them could lead to inaccurate results. Well, they're using underwater video cameras, so you might have a fish that's passed by multiple times, or not all fish would come within the viewpoint of it. They might went behind the camera when they're moving around. And that should pick up the mark for that one. Compare the distribution of blue and red male fish as depth increases. Well, as depth increases, the blue fish go down and the red fish increase for that. If they were now different species, they would no longer be able to interbreed to produce fertile offspring. Maybe DNA testing to see if there's big differences in their DNA counts, but it's the classic interbreed to produce fertile offspring, or do not do that. In this case, here we are looking at the phylogenetics, so we're looking at how closely related different species are. We need fossil records and sequence data, so the code laid out to compare. Last common ancestor of Amborella and Osmunda. All I'm going to do here is track them back. So I'm going to run it up here, along, down, along. And then for the Osmunda, so I'm looking for the point at which they are both at the same place. The last time, so we're going back in time from today. We're getting to this line here. I want to run that down. So we're at this point here. We need to figure out how much is each little box. Well, there are 10 boxes between 0 and 100. That must mean we're going up in tens. So at this point here, it would be 380 million years ago. I've already said the millions part 4 is, so 380. So track it back until they're both on the same, and this set up the vertical line. It's a common ancestor. How many groups of land plants monocots shared an ancestor with 200 million years, I should say. So we want to go here. Let's run that up. They're asking us, monocots. So monocots are here. So at this point, for the monocots, how many are also tracked back to there? Well, the eudicots do. Amborella does. Gymnospears don't, because they only match up with the monocot line later than that, or earlier than that, I should say, because we're going back in time. Hopefully you know what I mean. So that looks like just the two of them. There would be. If you had to name them, you'd have to name them. Agreed. Bit awkward here, because I think usually they've got them on the lines, if I remember correctly, but that's the way it is. That would be our answer. Multicoloured thing there. Monocots are more closely related to gymnosperms than Lycopodiophyta. Monocots, gymnosperms, Lyco. So if you look at the points where the blue and green meet together compared to yellow and green, that happens much closer to today. So the last common ancestor was more recent than the common ancestor of monocots and gymnospears. So monocots have a more recent common ancestor with the gymnospears than the Lycopodiophyta. That would work there. Genome is the entirety of the genetic information. All the coding, non-coding, and any kind of DNA we're dealing with. Don't know for certain whether you could say genetic information instead of hereditary, but passing on the information, things like that as well. Why can you get different enzymes from the single gene? Well, there is alternative RNA splicing, which is when different combinations of exons are retained. That's hopefully quite clear on this diagram, where in the first one, all 5 exons are retained. In the middle one, we've lost exon 3, and the last one, we lost exon 4. That changes your mRNA transcript, which then means you have a different protein formed from it. Moving on to our 4-mark questions, we're going to deal with the transcription one first. In this one, we're going from the DNA to the primary transcript. what can we pick up marks for? So where does it happen? It happens in the nucleus. The DNA needs to be unwound and unzipped. That's done by DNA polymerase. That's done by breaking the hydrogen bonds that are holding the strands together. Those are between the bases and what holding them together. And then you're going to form the primary transcript. That's when you need to deal with the complementary base parent. And here it's important to remember that the DNA will have the thymine and the mRNA will have uracil instead. Now, that should be enough to get 4 things. Talked about where it happens, what it does, how it works, complementing, all that kind of stuff as well. Now, could we still feed in some bits about what happens next, which is RNA splicing, where the primary transcript is spliced to form the mature transcript? Whether that counts as its own stage rather than transcription, yeah, I'm not too sure, but that's hopefully enough for 4. The other option was for chromosome mutations, and there are 4 of them. So we have our TIDD, TID. So for single gene ones, it was SID, substitution, insertion, deletion. And then slightly awkwardly, there's an I and 2 Ds in the other one, but they are translocation, inversion this time. You have duplication, and it also has deletion. So they have one in common, deletion. They both have one with an I, but it's insertion versus inversion. Right, what are those all about? Let's go for T for translocation first. This is when you have information coming from a different chromosome. So you end up with a gene present that wasn't present in the original one, which is a little bit different. We'll talk to duplication in a second. I should maybe say what that one was. Right, next up. So I for inversion. So in this one, a segment of chromosome is rotated, flipped, reversed. So using my wonderful example, this is where instead of having A, B, C, D, some of them would appear in the reversed order. So then we'd have D, C, and B for that one. Now we're into the D ones. I went for, let's go duplication. So now the thing is going to get bigger, but we'll see copies of the original letters. So this is the one that I was comparing to the translocation. So this time we're talking about a partner chromosome, the homologous pair. So this time the letter that appears, the letter that's added was something that was already in there. And then last up, we've got deletion. I'm going to say this most simplistic one, we're removing a gene from a chromosome for that. And then here you're going to end up with a smaller than the original piece of it. And that should pick up the 4 marks there. Moving on, we're now into metabolic reactions. Breakdown is a catabolic reaction. So how I remember the difference is cats are more likely to break stuff than build stuff, and they do that to release energy. And then anabolic would require energy and build up things. What happens to the active site when threonine binds to it, and how does this increase the rate of reaction? Well, threonine is the substrate in the enzyme's active site. We're talking about the induced fit. So it changes shape to better fit around it. Now with my chemistry hat on, that's going to lower the activation energy. It's like the less chemistry way of saying that is that lowers the energy required to start the reaction. Shuffling on, we're now talking about the pathway being blocked. So this is leading me to think about feedback inhibition. Now, is I seen the final product? Yes, it is in the pathway. So our final product binds to the active site. That's going to save resources and energy. Think of it like a conveyor belt, or think of it like a process where you get people working one after another. One starts something, then the next one finishes, and goes on and goes on and goes on. If no one tells the person at the beginning that they have enough of what they're making, they will just continually churn out more. In this case, intermediate 1, intermediate A, then B, then C, then D. and that's unnecessary because the body has enough, in this case, isoleucine of the final product for that. So it prevents the wastage of resources and energy. In this one, why does the rate of reaction level off a high substrate concentration without an inhibitor? There's only so many active sites available to do the reaction. We have saturated it with our substrate. All of the enzymes are working at maximum capacity. There are no more active sites available. Non-competitive inhibitor. A non-competitive inhibitor binds somewhere that's not the active site, and it changes the shape of the active site, which prevents the substrate from binding. In competitive inhibitor, they're both the inhibitor and the substrate are trying to get to the active site. If you have more substrate, they have a combined bigger chance of reaching the active site before the inhibitor blocks it. That's not what happens here. Increasing the substrate concentration has not reversed the effect of the inhibitor. So in this one, it is not reversed by high substrate concentration. So I'd put there. Here we're on to electron transport chain in respiration. Okay, exact locations. Well, we have 3 stages. Glycolysis is cytoplasm. You then have the citric acid cycle. And how I remember which one that is, that's the matrix. So M middle matrix of mitochondria. And then electron transport chain is the inner membrane of the mitochondria. That. Now think about it, if you look at it, we've got a membrane. Yeah, the phospholipids going together. Anywho, enzyme, what would enzyme doing? Enzyme is making ATP. Classic, lovely thing. It's making ATP, that is ATP synthase. Lovely stuff. Next, molecules Y and Z. Well, what's Y doing? Y is joining on to hydrogen. And we need to remember that we need oxygen at this stage. Oxygen plus hydrogen makes water. We should have our names for that. Slight chemistry rant here. I think if you write O2 and H2O, I don't think you should get the mark because it says name and that's not its name, that's its formula. But the marking scheme says you can have both of them. But in my opinion, which is right, shouldn't. Oxygen, water are what we have there. What is the job of NAD? NAD carries hydrogen ions and electrons from earlier stages to the electron transport chain. Next one. So a disease has all the structure of the carrier proteins. So what are the carrier proteins doing? They are helping things move through them. And that's looking like they're involved in so the electrons are moving around them. They're flowing through. Less ATP. Well, ATP needs hydrogen to flow through. For ATP to be made, hydrogen is flowing back through the ATP synthase. So if there's less electrons moving, there's less energy, less pumping across the membrane, less flowing back through, less ATP being made. For that, how much do I need to say for 2 marks? Let's go for less electrons moving through. Nope, let's go along, because along left to right rather than through. Along membrane, providing energy to pump hydrogen across. Less flow hydrogen through ATP synthase, therefore less ATP made. Whether we need all of them, I'm not sure. Paul went over the top, but that's kind of thing where the carriers are dealing with, for in this area here, electrons are flowing along the membrane, releasing energy, pumped hydrogen across, makes its way back through, spinning up ATP synthase and making it work. For that one, here we're talking about blood lactate levels. I'm just going to mark on the 2 parts we're talking about. So lactate is the dashed line, as we can see on the right. So when we come up to our line, we have to get the right, make our way across to the right. And then let's just grab the other one. 4.8. Oh, it's right then. That would make life easier. But again, we do need to go over to this side. So I'm just going to note down those numbers so I can scribble down when we need to. Each little box must be 0.2, 0.4, 0.6, 0.81. So we are going from 1.4 up to 6 point. Now, if I just move that around. Well, that's halfway between. So 6.2 and 6.4, 6.3. Let's go 6.3. Okay, so what's happening at first? Well, first we have a flat section or a section where it remains constant. So between 2.0 and 2.4 meters per second, lactate concentration stays constant at 1.4. And then I want to set up the next section. So that is between 2. Made a mistake like that. I wrote 2.5 and it should have said 2.4. So between 2.4 and the end of the graph, it's 4.8 meters per second. Lactate concentration increases up to 6.3. I've just realized I never put my units in the first value. I'll make sure to that here. That's 6.3 millimoles per liter. Like so. Next one, state the blood lactate concentration when heart rate was 135. So in this one, we're using both the lines together. So first job is heart rate 135. That will be halfway between, that will be here. Importantly, we need to go to the full line with the cross because that's what that left-hand axis links up to. Now at this point here, the temptation, and I am always tempted to just run it straight across, boom, 3.5. Not quite, because you cannot go from the heart rate line to the lactate axis. What we have to do is we want to go down. Now, technically what we're doing is we're actually saying, well, what speed does that happen at? That happens at 2.8. And then what we can do is go, well, if that's 2.8, well then at 2.8, we can work out the blood glucose, the blood lactate concentration. To kind of cut out that middle section, if we just then go down to that line, and then we want to run that across, and then we want to work out where we are sitting. So it would be 1.2, 0.4, 0.6. It's halfway between 0.6 and 0.7. That's 0.6 and 0.8, I should say. It is 0.7. 1.7. That's what I'm trying to waffle on about. 1.7 millimolar per litre. Millimoles per litre. Percentage increase in heart rate from 2 to 4.8. We are going to do difference over original times 100. For that, right, at 2. So starting point, we are heart rating at 122. So this section here is 122. And we are increasing by the end of it to 180. Remember, we need to go over to this side to work that out. If I could draw a straight line. Like so. So 180. Right, difference taken away from each other, you end up with 58. So that is the difference. The original, well, it was originally 122. Times that by 100 will give us, we go one decimal place, 47.5% as our answer. Now this one was quite, I would say, straightforward because we're going from 2 up to 4.8 meters per second, and then your values are going up as well. Some things it's a wee bit more different. You might say like, oh, compare this number to that number. One thing that can help is if you're in increase, you've got to start at the smaller result and go to the higher result. So if you make sure that your values that you're going to compare are in the correct order, so small to big if increase, big to small if decrease, then that will hopefully help keep everything straight forward, and then use your calculator to get that. Next up, we are asked for the average increase in power generated as heart rate increased from 84 to 164. Okay, now the units are going to help me here, hopefully, where it's watts per means divide, and then it's by your heart rate. But crucially, it is to do with increases of it. Now, I think we can do it where we just look at the biggest and the smallest, where it's gone up by 64, and then that has been caused by an 80 in the heart rate. So then we would have the watts is 64 of a difference. That's taking 80 80 beats per minute increase, and that will give you 0.8 beats per 0.8 watts per beat per minute. Sounds so weird to say. I was almost going to apologise thinking chemistry there, but in chemistry, anyone who's lucky enough to have done that, the average rate is changing quantity over changing time. Now, our top part is the watts, so it's the change in power, 64, over the change in, well, it's not time in this case, it's the heart rate is going to be for that one. And that has got me my answer there. There's maybe, I think there are other ways of doing it where you could, if you work out all the individual differences in power and in heart rate, then take the averages, so the average power increase and average heart rate increase, and then divide. The method I would use, start to finish, divide them, boom, sorted. Information from the graph state the athlete's blood lactate concentration when they generated 184 watts. Well, 184 watts was 164 of the heart rate. So 164 heart rate is going to be 2 boxes up. Heart rate goes to the full bar. Then want to go down to lactate, run it across, 2 boxes up. Each box was 0.2, 4.2, 4.4 should be the answer there. So it's just about taking your time, watch out for the fact that there's 2 axes involved, and try and get them in the right order. Cells that produce lactate. Oh, not the cells. I don't know the cells that produce lactate. The cell process that produces lactate is fermentation. Back in the day, anaerobic respiration, but fermentation's the expected answer here. Probably expected to spell it correctly as well. Next one, blood lactate. Okay, so we have a value that's decreasing over time, and that's because lactate is a reversible process where pyruvate is converted into lactate in the absence of water. If there is oxygen, as when they've stopped running and using up as much oxygen, that then is reversible for that. So I would say something like lactate is converted, turned into back to pyruvate would be the answer I would put. More graphs, lovely. We have got contaminated soils, bacteria. Okay, simple whole number ratio, carotenoid content, plants in each of them. Okay, so let's make sure we're using the right one. Carotenoid is the slashed one, so I don't care about that and that for the moment. Arsenic, there's only one axis, so it is these numbers here. And I make sure I'm putting them in the right order here. So 0.4, 0. Okay. This one's a wee bit tricky. If you're ever unsure, we can say, well, it's 0.2 of a difference. There's 5 baby boxes. That is 0.04 of a difference. So we would be 0.04, 0.08, 0.12. And then the last one is, well, if that's 0.4, that will be 0.36, and then 0.32 it should be. 32. I'm trying to put a wee 0 in here just to make them lined up in the same way. Couple of options here. Okay, now these numbers are a wee bit unhelpful, I'd say. A wee bit bigger than I might have wanted. So, could it be an idea to times them all by 100 to get them into like normal numbers? So 40, 12, and 32. They can all be divided by 4 to give 10, 3, 40 would be 10, so 8 of them. Is there any other number that can divide by them? That's looking like actually as far as we can go. So 10, 3, 8 for that one. You've got your calculator, so you can try out different numbers that can divide into each of them. One thing we could have done is divided them all by like 2 first of all. So 0.2, 0.06, 0. Like there are different ways of getting it. I'm just not sure if there's one way that just makes life easier. If you weren't catching that they all can be divided by 4, what you could have done at this point here is divided by 2. And then you can see, oh, you can divide by 2 again. 3, and then 8, and you still get to that answer. Calculator should hopefully help, even if the number's quite hard to spot. That should kind of give it a wee bit of a guide towards it. Describe the evidence from the graph that suggests that not all arsenic has been broken down. Well, none of them said 0 on it. Oh, wait a minute. By not all broken down by the ARB. Describe the evidence graph that suggests that not all arsenic has been broken down by the ARB. Oh, a wee bit lost with this one. Evidence from the graph says not all arsenic has been broken down by the ARB. Oh, right, that's not the arsenic levels it's talking about for that. Is it that you've still got... Okay, I've had to look up the marking scheme there for this one because I'm a wee bit lost with it. So what they're saying we have to look at the plants grown in this bit compared to that one for that. Oh, now I get it. Right, that makes sense. Okay, so this is our baseline. With it now. So your baseline one, where there's no arsenic, no arsenic to stop the pigment from being made, is up at 1 and 0.4. If we compare this one to that one, it has not got the same values. It's still lower than the soil with no arsenic. So if the ARB had got rid of all arsenic, we should expect that these numbers would be back up to the non-arsenic levels, but it's not. It's still a little bit below that. It's better than the one with just arsenic added and nothing to take away the arsenic. Okay. That one got me there. So plants growing in soil with arsenic and ARB have lower pigment levels than with plants with no arsenic at all. Next one, we are asked what's the point, what's the use, why are they helpful? They carotenoids, they are accessory pigments. This means they can, the plant can absorb wavelengths of light that chlorophyll can't. So they pass that energy on to the chlorophyll to supplement the energy from the red and blue light that the chlorophyll did capture. How does that lead to production of it? This energy excites electrons those move through the electron transport chain, releasing their energy. And then that allows ATP synthase to make ATP. Why do weeds reduce productivity of the plant that we want? It's all about the competition. It's going to increase competition for resources. Water, light, space. Selective herbivores have a greater effect on some weeds compared to wheat plants. This is the idea of the broadleaf versus the thin leaf ones. Some weeds have wider leaves. And that's going to mean that it's going to absorb more of the herbicide, more of the chemical. Next up, let's calculate how many times greater 2020 compared to 1980. This goes back to what I was saying about making sure you have in the right order. So what two numbers we've got? So we've got 2020 herbicide resistance, so right hand side. So it's 480 versus 1980 was 8. So 8 compared to 480. So we're trying to work out 8 times what is going to give you 480. One way you can do that is just divide them. So 480 over 8 will give you 60. So 8 times 60 is going to give you 480. So it's 60 times bigger. That would work. There, why was there a decrease in global food security over the range of... could you say either of them? One of them is that the population has almost, no, not quite doubled. It's gone quite a big increase of them, but there is also weed species that are resistant to weed killer, so therefore they would outcompete, or they would compete against the crop and reduce the crop population. Could it be either or both? It's 2 marks, so both. So rising population, I was going to say that means there's a higher demand for food, and then more resistant crops, more resistant weeds, I mean lower crop yields. Crops being the bits that we want, the wheat or whatever it might be. Natural selection, okay. So herbicides eliminate non-resistant species. Resistant species, they're going to reproduce and pass on resistant genes to the next generation. Number 11, pesticides. Independent variable is what is changed, investigated by the person, and then the measurement that we take is going to be your dependent one. So they are changing the summethion concentration. This is where I probably should have read the question to see what the heck that is. Insecticide, okay. Concentration. If the table has been put together correctly, that should be the left-hand side, what you're changing, and then what you're looking at to see what effect our change has had. Something else. So we're thinking about other things that can have an impact on the experiment. So hatching success, well, the temperature of the water is something that can affect how well the new egg, or how well the eggs hatch or grow. pH as well. Some fish will perform better in more acidic water or more alkaline or very neutral pH of solution. Could another thing be, they're talking about fish as like a generalized thing. Different species of fish maybe would prefer different temperatures, concentrations, or even this insecticide might affect it in a different way from the other ones. Like it doesn't treat all, it won't affect all species of fish the same, but those are the classic temperature, pH, classic ones. Which one acts as a control? Well, that's going to be 0. Have we got a 0? Yes. What happens when there's not any insecticide added? So it allows us to see everything left alone. Here's what we usually get. And now we can definitively say that any concentration of the summethion has decreased the hatching success. So it kills off the tiger beetles, but it could also harm the hatching. Interesting. Oh, lovely. Line graph. Right, along the bottom goes the left-hand column. Summethion concentration, milligrams per litre. I'll just drag the table with me. See me going up and down. Okay, we need to go up to 1. Now, there are 10 boxes, it looks like. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5, 10. So, a nice wee straightforward number for us would be 0.1 each little box. So 0.1 all the way up. I can squeeze these in. What you should be looking to do is to get the even scale going up the way, and it has to take up at least half of the page, half of the graph paper, which I have managed to do. Hatching success next, and it's looking like going up in 10s would probably be a good idea because we're going almost to 100. So that would work out. And then each little box, that makes it 2. So that should make it reasonably straightforward to put our numbers in. and we take up over half the page. Okay, so let's then put in our numbers. Okay, at 0, we're up at 92, so that'll be 1 box up. And then 85 will be halfway between 80 and 90. 57, so 50, 52, 54, 56, so halfway, so 3.5 boxes up. And then 42 is going to sit on a line. And then 25 is going to be halfway between 20 and 30. And then it's going to stay the same out to the end. Okay, so just to double check, we want a cross or dot or circle, whatever you want to do, but I need that every 2 big boxes, or yeah, 2 big boxes. So yep. Double check numbers, 90, nope, it's 92. Failed at the first hurdle. For goodness sake. Right, so should be, right, so 92, yes. 85, yes, halfway between 50, 2, 4, 6, and 7, yes. 42, yes. 25, 25, boom. Sorted. Now we just need to do the dot to dot, join them all up, getting us to our finalized graph. So there, oh no, try again. So there. And just take your ruler, go dot to dot, flat at the end, boom, sorted. State a conclusion. Well, it affects it up to a point. Above 0.8, it's staying the same. So as chemical concentration, 0.8 milligrams per litre. Hatching success decreased. And above 0.8, success remained constant. Side note, I always struggle to fit these all in. Now, my writing's fairly big, but people need to read it. It's what am I doing wrong? Why is it? No, okay, next one. Apart from it killing off the non-target species, what else can happen? It can build up within the organisms. So we can have bio accumulation where the individual animal will have more and more of it building up within their systems, or because that creature then gets eaten by a bigger creature, you can have biomagnification when as you go up through the levels. Because let's say it's a fish, so fish take in a little bit, bigger fish eats multiples of those little fish, so they're going to get a bigger value, and then a bird or something will eat those fish, and then they'll be consuming a lot of that pesticide. That is called magnification. Beat my answers there. Moving on, 12. Calculate the difference in lowest and highest milk yield from individual goats in this investigation. Now it's just any goats? Or are they saying... Right. I'm reading that as potentially 2 ways. Are they meaning parent 1, largest, smallest, and then parent 2, the same? I think that's really... My first thought was, are we looking for just the biggest of all? So like this one over here for F1 is bigger. But then the smallest one is from parent breed 2. From there. Calculate a difference in the lowest and highest milk yield from individual goats. Right, I think I'm going back. I think I'm leaning back to biggest versus lowest. Let's see if I'm right. So this would be, each box is going to be 10. So that's going to make that 730. And then this would be 520. And that would give you so 730 take away 520 is 210. So that'll give you 210 for that one. State how the graph shows that the milk yield was measured for more than one goat of each breed. Well, you have error bars, you have a range of numbers. So that's telling us that we've got a difference between them. Next one. In this one, whenever you move away from an F1 hybrid, you are increasing the variability in the characteristics. So F1 hybrids would have the hybrid figure, so they would have an increased milk yield or whatever else they're measuring. F2 are genetically varied. An advantage of intensive farming, it's lower costs, higher profits. Why can parasites spread more rapidly? Well, they're all in close contact. More animals in close direct contact. Close direct contact. High activity levels is hysteria. Someone's hysterical. They cannot be calmed down. The opposite one, low, would be the apathy. Or that one. Next, parasites. Parasites benefit by causing harm, or while causing harm, by causing harm. So what do the parasites do? So fly off spring benefit from food while harming bee larvae by a loss of food. Flies get food that was supposed to be for them by stealing the bee larvae's food. Something like that. Lay the eggs and has a social bee living colonies. Whether there's a fly to lay eggs in a social beehive. Aha. Does not guard their eggs. Well, in a colony, there will. That's going to be the worker bees defending it. So worker bees defend a social beehive. So a lot less of a chance of the flies flying in and laying their eggs undetected. Most of them are... In these colonies, all they care about is the survival of shared genes. So it increases survival rates, survival chances of the shared genes. Next, talking about African wild dogs hunting in packs. That's going to be cooperative hunting, working together. Advantage not already measures. So they mentioned that there's more success in that. They can also take away or kill larger prey. That would be my answer there. Whoops. Next part, we've got a graph. Estimate population 2018. I might be able to cheat the system. So that's another 2 years. So if I grab a line here and I extend out the x-axis like that, and now it's been quite stable over the previous 2 years. So if I continue that on, and as I did that, some of my lines are all lined up decently. That'd give you 7.1. Could there be an option for going, well, we were at 9.5 there, and then we've dropped to 8.9. So that was 0.6 of a difference. And then if this is 8.3, we've dropped by another 0.6. So an 8.3 take away another 0.6 would be, so 2017 we would expect 7.7. And then one more year would be another 6.6 off that, would be 7.1. Does that feel easier than what I did? Maybe. But yeah, hopefully either of those ways would pick up the correct mark. Moving on. Habitat fragmentation. So there are bits of uninhabitable land for the animals. We could introduce or make corridors between them that is habitat corridors. And this is meaning that each of the separate wee groups can then come together. That increases the access to food, mates, and then they can start repopulating them. Finishing off, we have our 2 7-mark essay questions. We'll do the first one first, talking about adverse conditions. Mentioned some of these earlier with the what did I mention earlier? I mentioned the estivation, hibernation, and daily torpor. Now, I wouldn't want to talk about hibernation here because that's to do with avoiding them rather than surviving them. So we're talking about types of dormancy. So dormancy is when the metabolic rate is reduced. These can be predictive, so before the onset of the adverse condition, or consequential. Make sure we're saying it's the onset. It's not after the whole thing's been happening. It's after it started. Differences here. So hibernation is for low temperatures. Estivation is for drought and warm conditions. And daily torpor is a temporary thing that happens at periods throughout the day. And this usually happens in those animals with a very, very high metabolic rate. Right, I'm just going to compare that to our marking scheme. So surviving with dormancy, that's looking like that would be 1. Reduces metabolic rate. So it says, sorry, the survive adverse condition by using dormancy. Talked about that. We've talked about the thing, each of the 3 types reducing metabolic rate, which we've definitely got. I think that's 2. It seems a bit weird to get both of them. Oh no, that'd be the next one, wouldn't it? Okay, I think 1 for that bit is looking good. Predictive before. That'd be the second one. And then that would be a third of the separate ones. So 4th one for the bit about hibernation and things like that. Daily torpor. 1. Okay, so I've missed out on a couple bits. So I'm reading through this. I could have or should have said that this allows them to survive the adverse conditions by using. So animals survive adverse conditions by using dormancy. So I might get the mark for saying, I've not said it outright, maybe I should have said that. That's one of the ways they can do that. I've talked about the metabolic rates being reduced. What I've not talked about is how they'd actually go about doing that. And that can be done by lowering heart rate and breathing rate. And then the other thing they've got is that this conserves energy. So I never said anything about energy, but that's the whole point of it, because they can't go about their normal daily routines. This conserves saves. So as a first edit, I'd say 4 or 5 is what we're going for. I just didn't talk about the bits that are, there's a bit presumed knowledge about the whole point of it is they can't just live how they would. So they wouldn't hibernate when it's warm enough for them and they've got enough food and things like that as well. Maybe a wee bit of tweaking I needed there. Growths of the microorganisms. That's going to be your lag, log, stationary, and death phases. Okay, so let's talk through these different phases. So lag phase is when the enzymes are being induced, so everything's getting ready. Log phase has very rapid growth. And this is because there's loads of nutrients for them and there's not that much competition. To full. So stationary phase is happening when food nutrients are starting to run out. And then also you have your waste products are accumulating. And in death phase, the cells will die due to lack of nutrients and build-up of toxic metabolites, so things that are being produced, waste that's going to kill them off. There are different types of counts that we can do, viable and total. So viable cell count is the living ones. And then your total ones are just everything dead and alive. Okay, let's compare that to the marking scheme. So we get 1 mark for naming all 4 stages. Done. That's just kind of throughout the course. Lag phase, enzymes are induced. Yes. Log most growth, very rapid growth. I've also got it in my graph as well, so I'm going to take that mark there. 4, plentiful supply of nutrients. Got that. Stationary nutrients are used up. They're running out. I'd say that as well. And or so we get 1 for that. It says that, or the tox things are building up. For that one, death, toxic accumulation of metabolites, nutrients have ran out, lack of nutrients build up. That sounds like the same sort of thing. And then they're giving you a mark for either viable or total viable living microorganisms. I've said that. Total estimates all cells dead and alive. That would give you that one. Other things to talk about are the secondary metabolites, antibiotics. So in this one, antibiotics are made which reduce the competition with other microorganisms. But I've got enough there that we pick up the marks. But for completion's sake, let's add that in. Antibiotics. So they're produced during the stationary phase and reduce competition with other microorganisms. And that is rounding that one off. That brings me to the end of the question and the end of the paper. Thanks for watching. Goodbye.